Welcome to the 2021 Edinburgh Parkinson's Lecture. I'm David Melton and I have the privilege of organising the lecture for the Edinburgh branch of the charity Parkinson's UK. It's a special year for us. It's the 10th anniversary of the establishment of our lecture. When I took over the organisation 3 years ago, I was told that one thing I must do was to make sure that I got back for the 10th anniversary lecture the person who delivered the inaugural lecture in 2012. I've succeeded, partially. Professor Roger Barker will be giving this year's lecture live because we're doing it online. We didn't feel able to go ahead with an in-person event again in Edinburgh this year because of the continuing coronavirus pandemic. In a minute, you'll be hearing from Mr. Patrick Mark. Patrick is the current vice chair of the Edinburgh branch, but he was involved in the establishment of the original lecture. Then, Professor Barker will be introduced by Dr. David Breen. David is a senior clinical research fellow at the Centre for Clinical Brain Sciences, University of Edinburgh. He's also an honorary consultant neurologist for NHS Lothian, based at the Anne Rolling Regenerative Neurology Clinic. Before we move on with the programme, I'd like to say a little bit about the question and answer session, which will follow the lecture again this year. We're keen to get as many questions as we can from people directly affected by Parkinson's. You can ask a question using the Slido box to the right of the screen. You may have tried it out during our test stream that we ran last week. You don't have to put your name on a question if you don't want to. But if you are somebody directly affected by Parkinson's, please type PWP into the name box. That will enable my colleague, Dr. Thilo Kunath, who's chairing the question and answer session, to identify the questions we most want asked. Just type your question into the box and click send. Even if you don't ask a question yourself, you can still vote for other people's questions. If you see a question you like, vote for it. That will boost that question further up the priority list and make it easier for Thilo to spot the most popular questions. I hope you enjoy this year's Edinburgh Parkinson's Lecture. Over to Patrick.
In 2010, I was chairman of Edinburgh branch of Parkinson's UK, and with a group of branch members attended the World Parkinson's Congress held in Glasgow. This huge event had a big influence on us, and we all considered Roger Barker of Cambridge to be the outstanding speaker. It raised our awareness of Parkinson's research, and we responded in 2011 by forming the Edinburgh Research Interest Group with Professor Ken Bowler as its chairman. At the inaugural meeting of the group in July 2011, members discussed potential events and speakers, and there was a general wish to invite Roger to Edinburgh and thus enable more people to benefit from his presentation. It was obvious that this should be a prestige event, not just 30 people in a church hall, and Ken Bowler and I agreed to investigate and develop a plan. We envisaged the target audience as being branch members and all people affected by Parkinson's, but also more broadly clinical professionals, researchers, fundraising donors and the general public. The aim was to raise awareness of Parkinson's, the urgent need for research, and Dr. Roger Barker's work. At the same time, we hoped to stimulate and encourage links between the branch, scientific researchers and clinical professionals. Key decisions were the date and the venue. If we were to attract professionals, it needed to be an early evening time, and for members it needed to be daylight, so April or May seemed sensible. As to a venue, it needed to be high quality and high capacity. We considered the Royal Society of Edinburgh and the Royal College of Surgeons, but both were too small, and we concluded the Royal College of Physicians of Edinburgh in Queen Street, with a 300 audience capacity, was ideal. Also, the Royal College was very helpful and welcoming, and we settled on 8th of May 2012. Organising such an event involves a huge amount of detailed planning, and we did ask ourselves, can we do it? Can we fill a 300-seat auditorium? We were encouraged by Lubna Arif of Parkinson's UK, who told us that Cambridge ran a similar annual lecture, and we thought, well, if Cambridge can do it, so can we. Ken Bowler drafted a multi-page checklist which went through many editions, but which guided us through the plan. Thilo Kunath an MRC research fellow in Edinburgh, was involved from the beginning. He suggested and arranged a linked seminar for Edinburgh professionals involved in treating or researching Parkinson's, which would generate a contribution to the costs. Other partners were Conor Maguire and Alison Stewart from the NHS Lothian Parkinson's Service and Catherine Crawford from Parkinson's UK. This formed our planning team. Ken and I worked on a draft publicity folder for the event, which could be mailed to branch members and others, and including the title of the Edinburgh Parkinson's lecture, on the spur of the moment we added 2012, just in case we wanted to repeat it. We both knew if we could pull off the first one, the next would be much easier, with the benefit of experience and the learning curve. On the night, the arrangements all went well, and it was generally considered to have been an incredibly inspirational evening. So it seemed obvious that we should use the experience and goodwill we had achieved to mount further annual events with world-famous Parkinson's presenters. When he spoke to us 10 years ago, Roger promised to come back in a few years' time with more and positive news. We have gladly taken up his offer and welcome him here tonight.
Ladies and gentlemen, hello and welcome. My name is Dave Breen and I'm a consultant neurologist and senior clinical research fellow based here at the University of Edinburgh. I'd like to join Patrick and David in welcoming you to this 10th annual Edinburgh Parkinson's lecture, which is one of the highlights of our Parkinson's educational program here in Edinburgh. And one of the reasons for that is that the event is organised solely by the local people affected by Parkinson's through the local Parkinson's UK Edinburgh branch and its research interest group. So I'd like to start by congratulating the branch in continuing to attract the highest calibre of speakers, and tonight's speaker is no exception. It's a great privilege to be here to introduce our speaker for this evening's lecture, who is Professor Roger Barker from Cambridge. Roger is Professor of Clinical Neurosciences based at the Cambridge Centre for Brain Repair and the Cambridge Stem Cell Institute at the University of Cambridge. He's also an honorary consultant neurologist at Addenbrooke Hospital in Cambridge. I've known Roger for over 10 years now, from the time that I spent my PhD with him down there. And rather than listing his various achievements and accolades, which you can read online, I'd like to just very quickly say 3 reasons why I think he's a perfect person to come back and give this 10th lecture. The first is that Roger's a great clinical and laboratory researcher and is very influential in both of those areas. His research has been important in better understanding the natural history of Parkinson's and the factors that influence how the condition changes over time. But also he's very influential in the field of novel therapeutics for Parkinson's, particularly the possibilities of stem cell transplantation in Parkinson's. And it's that topic that you're going to hear about tonight. But the second reason he's a great person to speak is that Roger is not just a researcher buried away in his laboratory or office. Roger's a great clinician, and he listens to people affected by the condition, and he uses that to influence his research. And so his research really is clinically and patient-focused. And finally, and perhaps most importantly and personally, Roger has a great knack of bringing people together through his personality. I've always been struck at how generous Roger is with his time when people come to him for advice and for his expertise, from the highest level researchers and other clinician colleagues, but also medical students, PhD students, patients and their families. And it's that bringing people together to try and improve care and treatment for Parkinson's, that is exactly what this lecture is all about. So without further waffle, I'm delighted to invite Professor Barker to give this evening's 10th annual Edinburgh Parkinson's lecture entitled Repairing the Brain in Parkinson's, Is This Possible? Roger, thanks a lot.
Well, thank you very much. I hope I'm not going to disappoint people tonight, but I've actually slightly changed my topic. So I thought, given that it was, well, 9 years ago, but the 10th lecture, the first of these that I gave, I thought it'd be useful to give a little perspective on where I think things have gone in the last 10 years, which will involve about repairing the brain with cell-based therapies, but it will also be a slightly broader perspective on this. And I want to thank you very much for inviting me back. The first time I was there, I remember taking home a most fantastic bottle of whiskey, which I was given, I think, from the Whiskey Club for the dinner afterwards. So I'm sad that I can't be there in person. And I hope that this works. It's a slightly odd experience, as you can imagine, that I'm talking to my laptop in my office in the hospital. So this is going to be a little journey through where I think Parkinson's has gone in the last 10 years. And I'm going to end with where I think it will go in the next 10 years. So these, I think, are the sort of new ideas and developments since I spoke 10 years ago. Some of these are actually ideas that were around at the time, but I think they've sort of gained prominence and become more significant. And I'm going to go through each of these in turn. So the first, I think, is around the better understanding of the heterogeneity of Parkinson's, by which I mean understanding the different subtypes of Parkinson's disease. So how well are we able to distinguish different groups of Parkinson's disease? How can we use that, this stratification, to better understand what goes wrong in this condition? How can we better model it, and how can we use that to inform ourselves about clinical trials? And that's really the dominant theme through all of this talk, will be how we can take things forward into clinical trials to really make a difference with this condition. I think the second major area where there has been a sort of, I think, a sort of greater understanding is this idea that Parkinson's disease is much more of a systemic disease than purely a brain disease. And in that context, I think the role of inflammation, so your immune system and how you react to the disease process, as well as predisposing you in some ways to getting the condition, has become a much more prominent issue. And that obviously has very clear attractions therapeutically, because we have lots of drugs that we can try to tackle the immune system and inflammation. So I think that's a second major development in the last 10 years, has been that inflammation in Parkinson's has moved center stage as something that is critical to how the disease plays out. I think the third major development, and this has actually been driven particularly by the Cure Parkinson's Trust, is better ways to trial possible disease modifying therapies, and in particular drug repurposing, the idea that you take a drug that's already in clinical use for one indication and try it in Parkinson's on the basis that that drug may help in Parkinson's disease in a different way to how it was originally used. So some of you will know simvastatin, very widely used drug to treat cholesterol. There's some evidence it reduces inflammation in the brain. It's been used in a trial led by Camille Carroll down in Plymouth in Parkinson's. Didn't work to slow down the disease, but that's the sort of principle. And I think that's gaining prominence. And with that comes some challenges, which I think are also on the verge of being sort of, well, not solved, but definitely improved. The fourth major area, which was gaining prominence in 2012, but has rather swamped the field in the last 10 years, is this idea that the protein that lies at the heart of Parkinson's disease is protein alpha-synuclein, which we all have in our brain, prominent part of our brain, but goes wrong in Parkinson's for reasons that we don't understand, can spread from one site to another. So this is this idea that the disease begins at one place, spreads to another. When it spreads to another, it infects a cell, that seeds pathology, and so it moves on. And with this idea has been, is Parkinson's disease starting at one site and spreading through the brain, spreading through the body? And if so, can we stop that spread by using immune therapies to silence the passing of that protein from one cell to another? And that has become a major area of development in the last 10 years. And I'll talk briefly about that. And then the final thing, which David very kindly mentioned in his introduction, around advanced therapies. And I'm going to tell you where stem cell therapies have got to with respect to Parkinson's disease, and how we're on the cusp of a mass of trials around this, and how this has again moved from something that was a sort of interest for odd people like myself, but it's now become much more mainstream as an area which people are keen to invest in and see work for Parkinson's disease. So those are the five major areas which I'm going to sort of walk us through, which I think have been the developments and why they give us hope, and where I think this will then take us in the future in the treatment and management of this condition. There are other areas which I could talk about. That's why I've called it a personal perspective. There's obviously been a huge recognition that Parkinson's disease has a phase before it clinically presents with the tremor and the stiffness and the slowness, the so-called premotor prodromal Parkinson's disease. characterized by this acting out of dreams at night, REM sleep behavioral disorder, loss of smell and such like. And everything I talk about tonight may ultimately be transitioned into this earlier stage if we can accurately screen the population, knowing that if we screen them for some of these factors, they will definitely go on to develop Parkinson's disease at a given time, and therefore we can present it in much the same way as other screening programs are used in the UK for breast cancer, colon cancer and such like. And that, I think, is a sort of underpinning sort of theme of this, is that what has happened in cancer in my professional lifetime, which is 35 years, I think is going to happen in Parkinson's disease. This idea that when I started, if you had breast cancer, it was just one type of cancer, you got the same treatment. Now, if you have breast cancer, it's typed, there are different types, you have personalized treatment, different regimes depending on what type it is. And with that, it's not only become better treatments for individual patients, but the prognosis for these conditions has dramatically improved. So that's really the structure of my talk tonight. It's slightly different, as I say, to repairing the brain, but I will come back to repairing the brain at the end, and I'm always very happy to take questions at the end around that. So let's start with this first thing about disease stratification and modelling, and this goes back to work that we did at the turn of the century. So Tom Falterney, who was one of my very first clinical fellows, who many of you will know, now a very successful neurologist and professor of neurology in London. We set up a study which was designed, it's called the campaign study, I probably talked about this 10 years ago, which was designed to collect everyone with Parkinson's disease in the county of Cambridgeshire over a 2-year period and follow them till they pass on or withdraw from the study. So we are still following these people up 20 years after we originally saw them, and we then collect data on an annual basis. Obviously the numbers get smaller as the years go by, and we follow them up, and our last paper on this was the 13-year follow-up. You know, headline figures from this are that the average age you get Parkinson's disease is 70, which was sort of, that's what people thought but hadn't proven. It also showed that the mortality rate, so people with Parkinson's disease in the community, if they get Parkinson's disease, do not die any earlier than people who do not have Parkinson's disease. So the mortality rate in the Parkinson's population in Cambridgeshire, at least, is exactly the same as the mortality rate in people in Cambridgeshire without Parkinson's disease. So most people die with their Parkinson's disease, not of their Parkinson's disease, which I think is a useful, encouraging piece of information, which this trial has sort of, or this study has thrown up. Obviously, with any one study, you can never be absolutely certain that it's correct. So we have now replicated this and has been done in other bigger cohorts in other places, namely in Oxford in particular. There's been in the Oxford Discovery cohort and Proband led by Donald Grosset in Glasgow. So there have been other studies which have also looked at this, as well as our own studies which have taken this on in new incident community-based projects. But this data of just collecting everyone with Parkinson's disease, follow them over time, collecting the information and then trying to understand it, has led us, I would say, to make 4 main conclusions, or take the data and the information in 4 main ways, which I think has been very helpful in shaping how we think about Parkinson's disease, both now and as we go forward into this new era of more trials and more experimental therapies to slow down or even try and repair the brain in this condition. So the first of this is that you can actually use this information to sort of subtype patients, and then that gives you information which you can use for a prognosis in the clinic now. So this data can be used specifically for looking at patients now. And simplistically, we would say that patients sit at one or two ends of a spectrum. This is rather simplistic, but I think this is a useful way to think about it. There are clearly patients who are younger, so these typically patients in their 50s and 60s who present. They may have some subtle thinking deficits, but these patients actually progress rather slowly and have a rather benign form of the condition. In contrast, there's an older group of patients who are greater than 72 years of age, who have problems with their ability to do certain cognitive tasks, naming animals, drawing interlocking pentagons, so trying to put things together in space, who also actually now seem to have a higher incidence of mild to severe constipation. And if patients present with that at their first consultation, they tend to have a more malignant form of the disease and tend to do less well and go on to develop early problems with falls and early problems of a major cognitive type and a dementia. So we can instantly see from this data that actually Parkinson's covers a spectrum from people who have a relatively benign disease to people who have a much more malignant disease, which is what we knew already, but this has put some sort of structure on it. So we can now use some of these tests to decide when someone comes to clinic in the first place, how they're going to do. Now that is obviously of use clinically now, but it also has value for how we're going to actually design trials and stratify patients into clinical trials going forward. So using this information, we've obviously also tried to understand what is it that drives these abnormalities. So why is it that some patients do better than others? Now, age is obviously one issue, and I'll come back to that, but we've started to look at this with respect to genetic factors. And so there are a number of genetic factors that we have actually identified which seem to predict whether people will do well or not. So we can now, when we see people in clinic, we can collect clinical information. We say, we think this patient's going to do well. We think this patient's going to do less well. We can now take blood from them, and there are certain genetic abnormalities or variations rather, which seem to predict that people are going to do less well. So one of these is a mutation in a gene called GBA, ApoE, which is a gene which is very much associated with Alzheimer's disease. And then this recent paper from this year has also identified other genetic loci, genetic areas, which seem to make people more prone to progress more quickly. So we're starting to understand why it is that some patients progress more quickly, and that gives us some insight into how we might be able to stop that from happening, and at the very least turn all the people who have a more malignant form of the disease into a more benign form of disease. So one of the factors which is driving it are these genetic variants which determine how cells handle proteins. Linked to that is also that some of those genes control your body's response in terms of the immune response and inflammation. And there's increasing evidence, as I've already said, that inflammation is a key determinant, not only of whether you get Parkinson's, but actually how quickly it progresses. So Caroline Williams-Gray, who actually works in the office next door to me, had this study and has taken this on, and I'll come on to this a bit later, showing that actually levels of inflammation in the blood when patients present, so if you take a bit of blood when they present, you can look at these genetic variants. You can also look at levels of inflammation, and people who have more inflammation in their blood when they present tend to progress more quickly. So inflammation somehow is associated with a more rapid progression of the disease. And the obvious third major factor, which I've already talked about, is age, which is one of the things that we find hardest to model in the lab, because most of the studies we do are with young cells that we grow or young animals. And as a way of trying to get around this, we embarked about 10 years ago now on a technology which tries to retain age using induced neurons. Now, I'm not a great basic scientist, but I know people who are, like Thilo, but this is Marlin Palmer and Janelle, a postdoc who worked with me and with Marlin. And between them, they came up with a strategy which enabled us to actually take patients' cells and turn them directly into nerve cells. So the idea here is you take a bit of skin, you grow the skin cells up, which are the fibroblasts, and then what normally happens is, in many of these studies, is you convert those fibroblasts into stem cells, so-called induced pluripotent stem cells, and I'll come back to that at the end when I'm talking about transplants, and then you turn those stem cells into dopamine cells. And those dopamine cells you make are obviously very healthy, but they're very young. And what we're interested in is how can we look at old dopamine cells? So what we wanted to do was to take the skin cells, and instead of turning them into stem cells and then into dopamine cells, we take skin cells and turn them directly into dopamine cells. And we know from other studies that people have done, as well as our own, that doing that retains age in those dopamine cells. So those cells now become old dopamine cells, which captures in some way age, which is clearly the biggest risk factor for getting Parkinson's disease. And so there's work which is ongoing at the moment. This is a complicated slide. You don't really need to worry about it. The point of it is that we can now do this reliably and make these so-called induced dopamine nerve cells, which actually have electrical activity, and they appear to have a pathology which varies depending on which type of patient we take from the clinic. So we're now starting to take patients and better understand what it is that's wrong with their, or could be unique to their dopamine cells, which means that they may require a specifically different treatment to people who have other defects, which we identify through this technology. So this is starting to open up the possibility of understanding why different people follow different courses with their Parkinson's, which in turn will give us insights into new therapies by which to treat them. The third major area in which we've used this data is to develop prognostic models. So the idea here is that we take the data, we plot it over time, and using this, we can predict when certain people or when people should hit certain milestones. So Jonathan Evans, who did his PhD with me about 10 years ago, did a lovely study where he looked at all the data and showed that the average time you took to get to certain stages of the condition. So in this particular graph, we can see at 46 months, at 4 years, on average, people tend to reach a period where they get Hernian stage 3, which is where they start to get a little problem with balance and falls. Now, this has use because we can start to think of this for clinical trials. We could have a disease-modifying therapy, for example, that we gave at the beginning of the disease, and we would say we would expect that most people at 46 months would be falling or having problems with balance. If at 46 months nobody had any problems, then you'd think I've actually probably made a difference to the natural history of this condition. So we can use milestones as a way of not only predicting where people will go and planning for the future for their care now, but also have implications for clinical trials. And Caroline has also done similar work using some of our data to make predictive models where you can actually put in certain factors around some of those tests I was telling you about at the beginning, certain abilities around balance, and you can use this to give people a score as to where you think they will go with their condition, and you can use that as a way of trying to predict as well as then use it as to stratify for your trials. And this is now coming back to what I said earlier, you can start to now factor in genetic information into these models so you can better predict where people are going with this. So all of this data enables us to now say, well, we can model where people will go with their Parkinson's disease. Different people will follow different trajectories. And we can use it to say, what are the key milestones? When are people likely to reach those? And how important are those? And this has led, I think, to a sort of new idea about how we can better think about Parkinson's disease. And Tom Fultony again and Camille Carroll, who I've already mentioned, have recently been in receipt of a large grant from the Edmund J. Safra Foundation, where they are in the process, which only began this year, of setting up across the UK the platform to do multi-arm, multi-stage trials, incorporating some of these models and milestones is the idea. So the idea here is that you can start running clinical trials with multiple drugs in pretty much everyone with Parkinson's disease at different stages, and actually use this to quickly move through disease-modifying therapies or putative disease-modifying therapies in a way which hopefully will change what we do. And this has been hugely successful in cancer. So the Stampede trial for prostate cancer has been running for nearly 20 years using this so-called MAMS approach. And the treatment of prostate cancer in 2021 is vastly different from when the trial was set up. And all of those changes have been brought through changes which have been seen in arms of treatment in people with prostate cancer. So some of this data which we generated around stratifying patients, predicting where they'll go, and these milestones is already starting to be thought of as a way of trying to take trials forward. And that is the sort of last area which I just want to pick up on this, that we've actually started to use this information ourselves to plan our own clinical trials. So whilst there are plans to try and develop these platforms for multi-arm, multi-stage trials, we've also used it for our own trials. And so just to illustrate this, the people and the patients who tend to have a more benign form of the condition, the group who are younger with these subtle changes, we would predict have a disease which is very much centered on the dopamine system. And as such, they would be very suitable for therapies which target the dopamine system. So dopamine cells, dopamine genes, they wouldn't necessarily be the best people to try disease-modifying therapies in because they progress rather slowly. So if you gave someone one of these drugs for 2 years, for example, which is a standard trial, you might not see much difference in 2 years. So actually, this group would be better probably for trialing cell and gene-based therapies. In contrast, the group who tend to do less well, the more malignant form, are probably the ideal group for looking for disease-modifying therapies, because we know when these patients will hit these sort of major milestones around falls and dementia, and we can start to use those to see whether we've made any difference with respect to modifying the disease. And we have used this strategy locally here in Cambridge to run 2 trials. The first is a trial that I'll come back to a bit later on, which is run by Caroline Williams-Gray, which is called AZA-PD. This is a trial in which a drug called azathioprine, which is an immune-suppressing drug, is being trialled in people with Parkinson's disease to see if it slows down the disease. And we have used the information we've collected in our longitudinal studies to say actually what the people we want in this trial are the patients who tend to have more inflammation at the beginning, who look as though they're going to do less well, and they will be the ones who will be the primary target for this therapy. In contrast with a trial that we've run, which was funded by the European Union, called TransEuro, where we were transplanting in fetal dopamine cells, so collecting dopamine cells from aborted fetuses, transplanting them into patients with Parkinson's disease. We selected patients who we thought would be in this good prognostic group, but have a disease which is very much centered on the dopamine system, who don't go on to develop major problems with falls and with dementia. And so this TransEuro study, which I'm not going to spend a huge amount talking about, this is the structure of it. It was a trial that began 10 years ago and is just coming to its conclusion. Essentially, we took the patients who we thought were in this good prognostic group, as we've said, we transplanted them, and we've just finished collecting the data on the last grafted patient 3 years after they had their transplant. So we grafted between 2015 and 18 and have just collected the data on that. I'll come back to that a bit later on in this talk. But for our discussion at the moment, the question I wanted to ask was, well, did we get it right? So the theory would be that we selected patients who we thought were in the good prognostic group. So over a 3-year period, for the run into this study, we would expect that they would progress a little bit, but not a lot. They wouldn't develop a dementia, and their disease wouldn't become very aggressive, and they require large doses of medication and develop involuntary movements and dyskinesias. So this was a paper we published a couple of years ago, and what we found was it was exactly as we had predicted. So in this group of patients, which was 100 odd patients collected across Europe, based on our model, we said they would progress slowly over time, and at the top graphs you can see that they progress very slowly with their Parkinson's disease in a linear fashion. So if we'd intervened with our dopamine therapy at that point and they hadn't progressed, we would know we'd made a difference. If you look at the bottom, which is this thing called the ACE, so that's the Addenbrooke's cognitive examination. If you're less than 84, you're in trouble. Anything above that, you're all right. They stayed exactly the same. The top score you can get is 100. So they did not develop any major thinking problems or dementia. And the graph next to it, E, which is AIMS around dyskinesias, the scores are well below 1, which is the lowest score you can get for any dyskinesias. So these patients behaved exactly as we predicted. And so this has given us confidence that we can use this natural history data to start thinking about trials going forward. And obviously that's informed this new initiative, this multi-arm, multi-stage trials that Tom and Camille are taking forward. So I think the first major development in Parkinson's disease in the last 10 years, which has really caught fire, is this idea of stratifying patients and putting selective groups of patients into selective trials rather than putting everybody into one trial. Now, the next major area, I think, is this role of inflammation in Parkinson's disease. So what is the evidence that there is a role for the immune system and inflammation in Parkinson's disease? Because up until fairly recently, most people would say, yeah, sure, there is a bit of inflammation in Parkinson's disease in the brain, but that's because cells are dying and they're unwell. And when anything dies and is unwell, the immune system has to come and mop it up. Just like if you cut yourself, you kill a few cells with the cut, along comes the immune system to actually clear everything up. So the immune system and inflammation is doing nothing more than just mopping up what's going on. It's not really involved with what's going wrong in the disease in the sort of key sense of it. So the first evidence that actually this was not true was a paper we had over 10 years ago now, where we looked at a thing called HLA. So HLA is very much associated, if it's associated with a disease, it tends to imply the disease has an immune basis. So autoimmune diseases like rheumatoid arthritis, psoriasis, inflammatory bowel disease, multiple sclerosis are all heavily associated with HLA because they're all autoimmune diseases. In other words, your immune system attacks some part of your body for reasons that is not clear, but nevertheless, that's a key event in how they come about. And we found that that actually was true for Parkinson's disease, that there was an association with this HLA. It was in a relatively small group of patients, but subsequently, in a paper the year after, this was validated and confirmed in a larger group of patients and has subsequently been confirmed further. So this means that HLA, which determines your immune response, is associated with Parkinson's disease, which means your immune system must in some way be implicated in the risk of getting Parkinson's disease. It's also, as I've already said, important in how Parkinson's disease then plays out once you've started. So this work by Caroline in our cohort of patients, so the campaign was the original one, Icicle was the follow-up study, same design. What Caroline found was that if people had inflammation in their blood, a pro-inflammatory profile, so they looked as though their blood was angrier, if you like, more activated, then actually when they first presented, if you followed those patients over 3 years, those patients who actually had more inflammation at the time they presented tend to develop quicker in terms of motor problems and tended to also do less well from a cognitive perspective. In other words, this would imply that the more active your immune system is when you present, the worse you're going to do over the next 3 years. It may change after 3 years from presentation, but that is what that data said. So this HLA association implies that the immune system in some way is related to getting Parkinson's. This study from Caroline would suggest that the level of inflammation at presentation determines how quickly you progress over the first 3 years of the condition. There's also some very interesting data, which is sort of slightly hidden, but is out there, which is a big study that was done in the United States in 2018. And what this showed was looking at large numbers of people in the population. It looked as though that patients that were taking actually immune suppressant drugs for whatever reason, whether it be for psoriasis, rheumatoid arthritis, or whatever, tended to have a lower risk of getting Parkinson's. In other words, if people in the population took these drugs, it seemed to reduce the chances that they would go on to get Parkinson's. Now, it may be, of course, the genetic risk of getting these other diseases is different to getting Parkinson's, but it's an interesting observation. And Caroline, in work which is yet to be published, looked at primary care data, and this is data from GPs, over a 20-year period. So this is significant numbers of patients, as you can see there. 64,000 immunosuppressed patients and 8.4. And in those who are on azathioprine, cyclosporin, such like, there was a 31% lower risk of getting Parkinson's if you were on those drugs. In other words, those drugs, at least in this study, would suggest that they may protect you from getting Parkinson's disease. So that again implies that they may relate, the immune system may be important in whether you get Parkinson's disease and then how you progress. And coming back to the original campaign study, so when we actually looked at this, we went back and looked at all the patients who were on non-steroidal anti-inflammatory drugs, so that's things like naproxen, brufen, aspirin. We also looked at people on steroids, so they were on this not for any reason to do with Parkinson's, but because they had a bit of arthritis or such like. And interestingly, we found that all the patients that were taking those drugs seemed to have a delayed onset to getting a dementia, as though these drugs in some way were preventing people from progressing at the same rate. Now, there may be other explanations for that, but all of that is rather suggestive that the immune system is not passive in Parkinson's disease, but is an integral part of it and is in some way driving what goes wrong in the condition as well as predisposing you to get it. Now, there's been further work on this. So Caroline's group has taken this on in the last 5 years. I can take no credit for this. This is all Caroline's work. So Rooney and Kirsten, who've been PhD students in her group, have actually taken this on, and they've shown, looking at different aspects of the immune system, so T cells as well as B cells. So these are all different types, and then cells which actually eat debris, monocytes, which are related to a cell in the brain called microglia, which tend to eat up dead cells and remove things which need to be removed, have all been shown to be abnormal in Parkinson's disease patients and tend to slightly segregate with the prognostic group. In other words, people who tend to have deficiencies in their immune system, which are quite subtle, or activation of aspects of their immune system, tend to sit in the group who do less well. So this implies that the immune system, again, is driving what's going wrong. And this has been taken on further by Nina, who's also been working on this, and also has shown in pathological specimens that in patients who develop a Parkinson's disease dementia, so these are people we followed in our study who died and very kindly donated their brains to our brain bank here, we find that in patients who have had and died with Parkinson's dementia, they have much more immune cells in their brain than patients who die with their Parkinson's and don't have such problems, which suggests again that the immune system is associated with dementia. It doesn't prove that it's driving it, but all of this is sort of circumstantial evidence that the immune system and your inflammatory response is in some way contributing to progression. Now, the only way you can really dissect this is by intervening. So I have to actually stop inflammation or silence the immune system in some way to see whether it makes a difference. Now, Caroline has put forward this hypothesis. So this is the what she's actually testing with her trial with the AZA-PD trial that I mentioned earlier. So the idea here is that obviously in Parkinson's disease, neurons die, so nerve cells die. That's probably intrinsic to the nerve cell in some way. And obviously the more nerve cells that die, the more the disease progresses. Now, microglia, which are cells in the brain, which are like immune cells, which live in the brain. So they're like macrophages or monocytes in the circulating system. These tend to eat things. They tend to therefore be important in clearing up debris and driving immune responses in the brain. If they're activated, they can also kill cells. And both nerves and microglia can be activated by the immune system when it's active systemically, so in your blood. So all of these speak to each other, and then the hypothesis is that in Parkinson's disease, for whatever reason, the alpha-synuclein, which we've already talked about, becomes abnormal. It starts to clog up the nerve cells and makes the nerve cells unwell, and they start to die off. As they die off, they release alpha-synuclein, which then activates the microglia, which then kill the nerve cells. The alpha-synuclein also diffuses into the circulation, activates the peripheral immune system, and so now we have a bit of a vicious circle. So as the cells start to die, the alpha-synuclein is released, that activates the microglia, that kills the cells. The alpha-synuclein activates the immune system systemically, and that now gets into your brain and kills more cells and activates the microglia more. And driving this may be the problems within the gut that we know now happen in Parkinson's disease. So there may be some leakiness in the gut, bacteria from the gut, and some of the toxins from the gut are getting into the circulation and contributing to this. So this all then drives a sort of vicious circle of increasing inflammation, driving the disease on. And therefore, if you could tackle or target one node in this system and silence it, then that may slow down the disease progression. An obvious place to do that would be by silencing aspects of the immune system in the circulation with drugs that we already have. And that's what Caroline's doing with this drug called azathioprine, which is a well-established drug which is widely used in neurology to treat conditions which have an immune basis, such as myasthenia and such like. So this is the hypothesis that she's testing in this AZA trial using patients that have a worse prognosis based on our disease stratification. So you can see that the big changes in 1 and 2 of the 5 points I've made are starting to come together in new trials, which offer real hope for patients because these are widely available drugs. They're not without side effects, but we can start to dissect the role of the immune system and inflammation, which I think is going to be a big area of research in the coming years. The third major development I want to talk about is this idea of drug repurposing. So the idea here, as I've said, is you take a drug that's in clinical use for one indication, blood pressure control, diabetes, and you now try it in Parkinson's disease patients, which of course short circuits huge amounts of development and time because it's already in clinical use. We know the side effect profile, we know the dose to use. It's very easy to translate it. Now, obviously, you could do that with any number of drugs, but you have to have some reason to believe that it will work in Parkinson's disease. So you have to look at preclinical data with cells of the type that Thiele makes, so stem cell derived dopamine cells or animal models of Parkinson's disease. And this, as I say, has really been taken on, I would say, most notably by the Cure Parkinson's Trust. And every year they have a thing called the Link Clinical Trials. The last one was this week, which I sadly couldn't go to. But every year they look at a portfolio of 30 to 40 drugs which are already in clinical use or are being used or being explored by pharma companies which have yet to be licensed but may be useful in Parkinson's disease. And over a 2 to 3 day period, we look at all the data that these drugs might be useful, both preclinical as well as there's any clinical evidence, and then one ranks them as to which drugs should be supported for clinical trials. And in the last 10 years, all of these drugs have now been put into clinical trials. So there are lots of drugs which work on diabetes, so-called GLP-1 agonists, of which exenatide is probably the most well-known. Isradipine, which is a blood pressure tablet, has been the subject of trial, and so on. So all of these drugs which have been in use for other indications have now been turned to be trialed in Parkinson's disease. And of course, the ambition will be that in the future, we can take 10 drugs or 5 drugs at least, and throw them into the MAMS trial with clear milestones and such like. So there is a hope that all of this process can be accelerated. And I just want to take you through the exenatide story, which I think has been a very interesting exercise in a sort of exciting new development around drug repurposing, which is currently at the sort of cusp of whether it's actually going to work or not. So this again has been taken on by Tom, tirelessly working away in London on this. He did a trial which he published in 2013, where he essentially took people with established Parkinson's disease, and they were either given exenatide, which is an injectable treatment, or they were given nothing. It wasn't that they had placebo, because for this trial, the placebo, an injectable placebo, is actually costs about £250,000. So actually, injectable placebo was much more expensive than the injectable drug itself. So what Tom did was he just said, well, we're going to take people, they'll be randomized, but they won't have placebo, and we followed them over time, and we treated them for 12 months. And what he found was that when you gave people exenatide over a 12-month period, their Parkinson's disease seemed to get a bit better with respect to their motor scores, and their thinking also improved. And those who just had nothing and were just being followed as part of their routine care actually got worse. He then stopped the treatment at 12 months, and at 14 months, he reassessed them. So they're now 2 months after they finished the treatment. And if it's disease modifying, you'd expect that these improvements would still be there. If it was simply making patients symptomatically better, then once you've stopped it, the symptoms would come back and they would go back to where they were. And that was not seen. So it implied that this drug may have some long-term benefits. And then a year later, he published a paper where he'd followed them up 12 months after he stopped the treatment, and saw that the advantages persisted. So all of this suggested that this drug may actually have some disease modifying effects. So this led him to then go on and do a double-blind placebo-controlled trial that he published 4 years ago, where he took patients and they now received either exenatide or something that looked like exenatide, which was a placebo. He followed them over 60 weeks, and at the end of 60 weeks, patients on exenatide seemed to be in a slightly better place than patients who had been on placebo. Now, it's a bit difficult to necessarily interpret this data because the improvements were all at the beginning, but it also at least supports the idea that this drug may have a disease-modifying therapy. Tom then explored the reason for this, and one of it possibilities is it may be actually influencing inflammation in the brain, coming back to the previous point that I made. Tom now has got funding from the National Institute of Health Research, where he's doing a 2-year study with 200 patients with exenatide, which is currently recruiting and should finish recruiting at the end of this year, the beginning of next. That's not the data. That's what he's hoping to show, that if you go over a longer period of time, the difference you will see will be magnified, and this will be shown to be truly disease modifying. So drug repurposing, I think, is really here to stay, and it's rapidly advancing how we can trial and give hope to people with Parkinson's and how we can actually make a difference to the natural history of this condition. The fourth major development has been this idea of protein spread. So the idea here, as I say, is that Parkinson's disease begins at one place, in the gut, in the nose, in some part of the brain, and from there it spreads in a very reproducible fashion from one site to another. And this goes back to really work that was done at the beginning of this century by Heiko Braak, a neuropathologist in Germany, who said that there are different stages of Parkinson's, and you can work out where people are by these stages based on the pathology, and those stages are linked by where the protein spreads. This then took on a whole new lease of life in 2008, when patients who had transplants of fetal dopamine cells, just as we were doing in the TransEuro study. Some of these patients died 10, 12, 15 years after they had the transplant. And what they found in those transplanted dopamine cells was the pathology you see in Parkinson's disease. And that was a bit odd, because those dopamine cells in those transplants, which have come from aborted fetuses, are only 10, 12 or 15 years old. And you don't see Parkinson's disease in people who are 10, 12 and 15 years old. So one of the theories was that the reason why this pathology had developed was that the protein, the alpha-synuclein, which aggregated in the Parkinson's brain to give them pathology, had spread from the Parkinson's brain into the transplant, seeded pathology there, and was causing Parkinson's disease to start to develop in these young dopamine cells. And on the back of that, a whole number of studies have been done. This is just a few, showing that if you inject alpha-synuclein or extracts of Parkinsonian brain or all sorts of any variant on that, you actually can spread pathology in animal models, which looks as though the protein itself can spread and can actually cause disease. So this has led to a whole new sort of idea that Parkinson's disease is a protein that goes wrong and then spreads from one site to another. And this has even led to a new classification. So Per Borghammer in Denmark has come up with the idea that there are two types of Parkinson's disease based on the spread. One is where it starts in the brain, stays in the brain, but might spread into the gut later on. So this is a disease that begins, say, through the olfactory, the smell system, and goes into the brain and later comes out to the gut. And then there's a body first version where the disease begins in the gut, spreads up to the lower part of the brain stem, affects areas there, and then eventually spreads up, affects the dopamine cells, and then further areas of the brain. So this is his idea that Parkinson's disease may have two different variants depending on how this protein spreads. Some are associated with this sleep disorder, this REM sleep behavioral disorder, and some are not. And that's how you can explain why some patients have it and some do not. And that he would therefore postulate that there are these two types of Parkinson's disease, which are based on protein spread as well as clinical features. And if you want to hear more about that, then I did a research spotlight with him a month or two ago on the WPC, where we spent an hour discussing this idea. This is not new, the problems with sleep, and Dave Breen, who was earlier on tonight, did lots of wonderful work around sleep and circadian rhythm problems in Parkinson's disease. So how that factors into all of this is an interesting area. But this, of course, raises the question, as I said at the beginning, that if the protein spreads from one cell to another, whether it comes in the brain and spreads through the brain or comes through the gut and spreads up into the brain, if it's passing from one cell to another, at some point it must be outside the cell. So could we actually kill it with an antibody? So just in the same way as if you've had COVID or you've had a vaccine for COVID, you will generate an antibody that will target the nasty bit of COVID and kill it off. And the idea here is you'll get an injection, a vaccine or an immune therapy, which gets you to produce an antibody which kills off the bit of alpha-synuclein that's driving the disease. So this has become a major pharma interest in the last decade. There are now multiple trials where people are looking at these antibody and immune therapies to silence alpha-synuclein in people with Parkinson's disease. And all we can probably say to date is that all of those therapies are safe, but whether they make a difference or not, we've yet to see. So that would be the fourth major area. I think we've first of all better understood how to subtype people with Parkinson's disease and understand why they follow different subtypes and how we can use that to better model disease and design trials. I think we've got a greater understanding that inflammation is critical to the development and progression of Parkinson's disease. I think we've understood that trying to repurpose drugs actually has great merit, and we might be able to do that very effectively and actually look at some of those earlier hypotheses that we've come up. And fourthly, this whole area of protein spread and immune therapies is an area which in the next 5 to 10 years will actually tell us whether this is an approach that has merit. And the final area I just want to talk about in the last few minutes is around actually cell-based therapies. So the idea here is that actually what we're going to do in Parkinson's disease is we're not really interested in why people get Parkinson's disease. We're not really interested in what makes people progress, except we just need to choose the right people for our trials. But essentially, we know if you lose 250,000 dopamine cells from the substantia nigra projecting up into the brain, if you've lost that number of cells, you develop features of Parkinson's disease. So if we put back 250,000 dopamine cells, in theory, we should be able to repair that bit of the brain and deal with that aspect of the disease. And that bit of the disease are all the bits of the disease which you currently have that respond to the therapies we use in the clinic at the moment. Now, what could we use to replace those dopamine cells? Well, as I've already said, you could actually get dopamine cells from the developing human fetus, which comes from aborted tissue, which obviously comes with major ethical as well as practical problems, as I'll come on to. You could actually make dopamine cells from, for example, skin cells, as I've already said. You turn them into, take skin cells, turn them into stem cells, turn them into dopamine cells, and then put them into the brain of patients with Parkinson's disease to replace those that are lost. Or you make dopamine cells from embryonic stem cells, which are the cells which come from spare embryos from IVF programs, which again you turn into dopamine cells and transplant into the brain. And this is not new. This has been around since the 1980s. And actually the data is quite encouraging in that when it works very well. So there were 2 patients who were in London. They went to Sweden, had a transplant in the early 1990s. They were followed up, subsequently died from causes unrelated to their Parkinson's disease. But essentially what this graph demonstrates is that they'd had Parkinson's for 10 years. They had a transplant, and for the next 20 years, their Parkinson's did not progress. So their motor scores 30 years after they presented were exactly the same as when they first went to see their doctor in the 1980s. So this showed that the transplants could stabilize Parkinson's and took them back to normal over a 30-year period. This was associated with normalization of dopamine on their scans. A patient in Lund who had had a very dramatic response to their transplant died 24 years after the cells were put in. And at post-mortem, although there was a bit of Parkinson's pathology in the transplant was still surviving, and the vast majority of the cells, 80 to 90% of them, actually were healthy dopamine cells. So fetal dopamine transplants have shown that you can get long-term clinical benefits and survival. The results have, however, been very mixed, and that was what led to the TransEuro trial that I already mentioned. Now, this was a trial not without its complication. We got funding in 2010. It took us to 2015 to graft our first patient. It took us 3 years to graft 11 patients. And as I say, the 3-year endpoint has now just passed, so we're collecting the final data on this. Now, this next slide is merely just to tell you all the problems we had with this trial, and there's one I want to come to at the end. So we had problems in getting the regulatory authorities to agree as to what this trial was, both within the UK and other countries. We had problems with some of the partners we had across Europe, another regulatory problem, a problem with the company who was managing our grant. Sadly, one of our major investigators died. Another one had to move on. We had problems with regulation in Sweden. We had unexpected breakdowns in equipment. We had problems with trying to understand what we needed in certain countries. We then had difficulty in whether we could reproduce our data across sites. The hospital here changed its policy, so we couldn't use the device we were planning to use. We then moved to an electronic hospital system, which meant all elective surgery was cancelled for several months, including our research surgery. We had problems with sterilising the instruments. Then they decided to rebuild our neurosurgical theatres. Then the junior doctors went on strike, which wasn't related to any of the above. So we had no junior doctors, so we couldn't bring patients into hospital for this type of work. By this time, we'd spent so long trying to get the trial going that all the reagents we had bought and made for it passed their validation date, so we had to make more. We had great difficulty getting tissue from one site to another, and that was before Brexit. We then left the EU, which has caused major complications with trying to get patients here from other places. Then COVID came along, which meant our long-term follow-up was an issue. But the critical problem that I was going to come to at the end, which was the one which really shows that this therapy has no merit going forward, is that we simply could not get enough tissue. So you need at least 3 to 4 fetuses to graft one patient. And in the 3 years we did this trial, we grafted 10.5 patients. One patient only had one side grafted. And for those 21 surgeries, we had another 100 odd, which were cancelled because we did not have sufficient tissue to do it. So this makes the point that if this cell-based therapy is ever going to go forward, we need a stem cell where we can reliably get the cells out of a freezer and treat patients in a reliable, predictable way with a standardized cell product. And that has become possible because just over 10 years ago, 2 groups, Lorenz Studer's group in New York and Marlene Palmer's group in Lund in Sweden, developed protocols which enabled us to take human embryonic stem cells and turn them into authentic dopamine cells of the type that are lost in Parkinson's disease. And on the back of that, we set up an initiative which brings together all the groups around the world, which first met in 2014 to try and coordinate this effort, so that rather than one team just try and do it on their own, we work together to make this work for everyone with Parkinson's disease. And so this has been a very successful initiative. We meet every year, apart from when COVID came along, to actually ensure that we can do this as quickly as possible. And just to give you some idea of how much this has changed, when we set up G-Force in 2014, the global investment in stem cell-based therapies for Parkinson's was approximately $0. Currently, I would say that the investment in this field is well in excess of $1 to $2 billion. So this has become a major interest for pharma. So we work, myself and Marlene Palmer in Lund with Novo Nordisk, the 4th biggest pharma company on the planet. Lorenz Studer and Vivian Tabar's group in New York work with Blue Rock, who are now owned by Bayer. There's a group in Chicago who now Fujifilm Cellular Dynamics who are working on this. There's a company called Aspen Neuroscience in California working on this. The group in Japan who invented iPS cells are working on this. The Mass General and Harvard also have a program around this. There are groups in China working on this, and there's a trial going on in Australia with a different type of stem cell. So all of these groups are now working on it. Where have we got to with this? Well, the group in Japan did their first transplant using an iPS-derived dopamine cell in 2018. They've now done at least 5 patients, or had by March of this year. So they are progressing with their trial. The group in Mass General in America published a paper last year who had their own cells. So they had autologous, i.e. they took their own skin cells, turned them into dopamine cells, and had them grafted in and showed it was safe with some effect. The group in New York have started their trial this summer with their ES-derived dopamine cell, and we are planning to start our trial in the first quarter of next year, and the documents are going to the regulatory authorities next week in Sweden, and shortly after to the MHRA in the UK thereafter. So to conclude, I think a lot has happened in the last years other than just COVID, which is what all we think about. So I would say that the 5 major areas which I've talked about, disease stratification and modelling, I think has now become much more the standard way in which we think about Parkinson's disease and its driving trial design. The role of inflammation as not a bystander but cause of the disease, I think, has become accepted, and that's leading to new ways to try and target the disease by slowing it down through that route. Drug repurposing has opened up all sorts of possibilities and has shown that it is easy to do and can be done, and is being done in large numbers of patients and trials. Protein therapies is really the, immune therapies is really the domain of the pharma, but that is now well established in terms of trials. So that will be very interesting, exciting to see what comes out of that in the next few years. And dopamine cell therapies, 10 years ago, I was really on my own with that, and now everyone thinks that's a good place to go. And so, as I say, this may all shift to early disease. So what do I think in 10 years' time? Well, I probably won't come back in 10 years' time because I'll be so old and decrepit. But I think actually we will refine this modelling and stratification. We will know more about genetics. We can now get more out of post-mortem analysis of brains. So I think we're just going to get better and better at refining and categorising people with Parkinson's in the way that cancer has done that over the last 20 years. I think we're going to be clever about doing design trials with these multi-agents and multi-arms, and I also am a great fan of polypills, giving people lots of these drug repurposing agents to see if we can target all pathways and slow things down rather than use one at a time. I think we're going to be much smarter about endpoints, so rather than just sort of having slightly unuseful ones, we're going to ask patient-related outcomes, what actually matters to you, and use milestones. So when do people fall? When do they develop dementia? Things which are very concrete, and I think will give a much greater confidence that we've made a difference. And I think, and I hope, that dopamine cell and gene therapies will become something more than just a trial agent, but will actually start to become a therapy which people will be offered, not only at the point at which they now get DBS, but could even be first-line therapy if the trials really show that they work very effectively. And so with that, I would like to finish. I would like to thank all of the people who've helped me over the years. The group at the top is in a pre-COVID, non-socially distanced lab meeting from February of 2019. And the group below is the STEM PD, our STEM trial group between Lund and Cambridge in January of last year. And with that, I would like to thank you, thank all my funders. Thank you very much.
Thank you, Roger. We'll give you a second to catch your breath, and we hope you're ready for lots of questions, because Thilo's got a lot of questions for you. Go ahead, Thilo.
Oh, great. Hi, everyone. I hope you can hear me. And thank you, Roger, for an incredibly comprehensive and inspiring lecture. And one of the questions that we may not get to is about how do you keep going with all these problems that you're facing, and you're so resilient. So I'm very, very pleased that you're our lecturer this year. Okay, I'm going to start at the very top. The most popular question is about food. Is there any known food that is beneficial or that retards the progress of Parkinson's, or any foods that should be avoided?
Good question. The simple answer is no. There is nothing known on it, but that doesn't mean that there isn't something that could modify the disease. So there's a great deal of interest about the bacteria in the gut, which I didn't talk about, the microbiome. And obviously the bacteria you have in your gut make up a large part of your, well, they actually weigh quite a lot, and they determine quite a lot about disease states and disease progression. The question is, how do you influence that? So probiotics is what people have been quite interested in, but and there's some sort of preclinical data on that, but there's nothing that's been shown in the clinic. In terms of diet and food, there's nothing in particular that one knows about that influences the progression of Parkinson's disease. It's obviously well established that certain foods will influence the uptake of drugs, so protein sparing diets has been around for a long time because some proteins compete with dopamine to get L-dopa to get across the gut. So sometimes people change their diet because of that. So there's nothing that's been shown. That's not to say that something might not work. Personally, I think there's more likelihood that drugs are going to do it than diet.
Okay, great, thank you. Actually, this is a highlighted question from David. I'm going to summarize it. Do you think it's a pipe dream to start banking our own personal iPS cells now for future neurological transplantations such as you just described? Should we be doing this now, or is it too early?
Yeah, so this is an interesting question. So one of the questions is, you know, should you use the patient's own cells to make their dopamine cells? So Tilo's got Parkinson's. I take a bit of Tilo's skin, I give it to Tilo and say, turn it into a dopamine cell for me, and I'll put it back in your brain. So the reason you couldn't do that at the moment is that the regulation is such that I'd have to do all the necessary testing on Tilo cells, which would cost probably several £1 million, and ultimately I can only treat Tilo. So that's not very economically sensible. There's also a theoretical problem that Tilo's got Parkinson's disease in this scenario, and I'm now putting back dopamine cells from him that have already declared themselves to have a predisposition to getting Parkinson's. So your dopamine cells might not be the best cells to put back in your brain. So I think autologous grafting has practical and theoretical problems. Now, there is moves afoot, particularly in Japan, to say, actually, let's look at all the immune subtypes of people in Japan, so the HLA subtypes, and let's make a bank which covers all of those subtypes, so that when Tilo develops Parkinson's, I can type his immune system or his HLA and say, you've got this type. I go to my bank. This cell here is very well matched to you, so I'm going to give it to you, so you need less immune suppression. And obviously the advantage of that is not only could I then treat Tilo's Parkinson's, but I could then treat his diabetes if I can make islet cells from that. I could treat his heart disease by turning him into cardiac cells. So people are very interested in this idea of banks. In answer to the specific question, I would say no, don't do it now, because I don't think there's enough evidence for that, and there's not enough infrastructure and support to allow that to happen, because you can't just make the cells any fashion. You have to make them to a clinical grade, and that is expensive, and that requires specialist places to do it.
I would agree with that as well. I'm going to combine the next two, and a bit related to your last slide. How far are we away from getting a cure, and what are the biggest developments in the next 3 to 5 years?
Yeah, so the question is, so obviously we would love a cure. That would be a good thing. But actually, if you can slow the disease down by 50%, you're probably almost cured it. So let's say the average age to get it is say 70, and after 15 years it becomes quite difficult to control. Well, if you slow that down by 50%, that's 30 years. So you then start to run into problems when you're 100. I suspect other things may have overtaken you when you're 100. But if we then say, well, actually, we can pick Parkinson's up earlier when you're 60, 10 years before you normally present, and then we slow it down by, now it's 25 years till you get problems, but now we slow it down by 50. Now you're up to 110. And so there'll be other problems that are going to be in your way or not here at all. So I think slowing down the disease by 50% is not an unreasonable aim. I think the other thing is actually the disease progresses quite slowly in pretty much in most people, which means that you're quite good at handling the pathology. So actually, I think whilst the pathology obviously evolves and eventually overwhelms the cells and causes problems, you're quite good at handling it. So I think the tweaks that we need to make the cells better are not going to be huge. So it may be small changes actually make a big difference, or make us enough difference. That's
It sounds realistic and achievable when you describe it like that. I should have mentioned when you please upvote questions as we're going, because there are about 40 and we're not going to be able to get to all of them. So the next one is, can CBD cannabis help during off periods, or does it interfere with medications like L-dopa?
Yeah, well, I don't know enough about the pharmacology of it. What I would say about cannabis or CBD is that obviously in your brain you have cannabis receptors, and they are also in the area where the dopamine normally works. So people have been quite interested in using cannabis-like drugs to try and treat, in particular, dyskinesias. John Brocci and Susan Fox used to do a lot of work on this. My own experience from what patients have told me is that in some people cannabis is quite helpful, and it may be quite helpful because it calms them down. So as everyone will know, if you're very anxious or stressed, that makes your Parkinson's worse, it makes your dyskinesias worse. So some people find that cannabis oil, and some people cannabis, not that I would advocate that of course, actually helps, and I think it may help because it calms people down. I don't really think it interferes with L-DOPA per se, but I'm not an expert on that, so I would have to go and look it up. But that's not been what people have told me. It's been more of help in making people more relaxed.
Less anxious, yeah. I think it's legalized now in Canada, where I'm from. So the next question is a more general one. What are the top 5 tips for people with Parkinson's? Very popular question.
Top tips. Okay.
Top 5.
So be positive. Be positive and take control of your condition. Don't let your condition take control of you. So you need to take control of it, and you need to decide what you want to do. And the job of the doctor and the team around them is to make you do what you want to do for as long as you can. So I think people often just slip and say, well, I'm a bit older, I can't be expected to do this or that. I think you should be much more proactive and positive about it. That's the first thing. Second thing is get lots of exercise. So I think being physically fit is very good. So if you just sit in the chair and do nothing, you'll get unfit and walk less distance, regardless whether you've got Parkinson's or not. And there is sort of evidence, at least preclinically, that exercise produces fertilizer type substances in the brain, produces new nerve cells. This is in rodents. I don't know what it means for people, but it sort of implies that being active is good for your brain. So I would take control of your illness. Don't accept that you can't do more about it, but try and work with your medical team to get the most out of it. Do lots of exercise. Get yourself into some research clinic, and make sure you link up to the charities and such like, so that you're aware of what's going on. So I think being very proactive is what I would advocate, and be very wary of anything where people offer you a therapy which you've not heard of and you have to pay for, because basically any therapy, if it's got any proven efficacy, you will not have to pay for.
Yeah, great answer. I think Alison Williams would agree with your answer. Next question is, we take our medication, Cinemet, regularly as advised, but our body's demand for dopamine fluctuates throughout the day. Is there any way this can be reconciled?
So that's quite common. So again, it comes back a bit to this sort of different types of Parkinson's. So one of the curious things about Parkinson's is it's very common for people to have good time. So in the early stages, people are pretty good all the time. But as the condition evolves, people find that they have better times and worse times. So some people find the morning is worse before they take their medication. So they when they wake up, they're dreadful, get their tablets and they're off. Some people get huge sleep benefits. So they actually say, I'm absolutely crackerjack first thing in the morning, do very well. It's actually when I take your tablets that I start going downhill. I think it's just because they're running out. Most people at the end of the day are normally worse than they are in the middle of the day. And that's probably because we're just trying to guess how much dopamine to put into your brain at a certain time. So dopamine is very finely regulated in normal brain. You get sort of phasic releases. In other words, you get bursts of it, and then you get this sort of slow release. And we're just giving tablets which basically flood the system and hope we get enough in enough time. So that's why it's very difficult as people evolve to get the right combination. You can often do much better than we sometimes think we can by splitting doses, but then you have to just be a bit wary that your whole day isn't taken up by taking tablets every 45 minutes or 1 hour 15 minutes or whatever. But there are ways, once you get beyond a certain point, where you can infuse dopamine in a much more continuous fashion, duodopa. There's going to be a subcutaneous, I think, form of dopamine that looking very promising, which might come along. So I think it's not unexpected, and part of it is just the slightly sledgehammer approach we use at the moment for treating it. And that's of course why cells are quite attractive, because if we put dopamine nerve cells back in, they will release dopamine in a more normal fashion all of the time.
Great, thank you. Very good answer. This is another question. Are there any techniques for reducing tremor that does not involve medication or deep brain stimulation?
Yeah, so people have always been quite interested in this. So there are some various bracelets that people have developed, I think, for this. So there used to be, when I was growing up, there used to be a treatment for tremor. It used to be put weights on your wrists. And the obvious answer to this is you'll end up stop shaking because you simply can't lift your arm up because they're so blooming heavy. But so some people have tried still using that for damping. I think there are people who've developed devices where they try and use sort of biofeedback from your tremor to feed back to try and suppress the tremor. I mean, I don't know much about it, but that has certainly been looked at as a way of trying to control tremor in a non-pharmacological way.
I was wondering if they were referring to focused ultrasound. I don't know.
Well, focused ultrasound is obviously, that's brain surgery really, and that's so that's I mean, it's a good point, Tilo. So I mean, outside deep brain stimulation, there is this thing now called MRI focused ultrasound therapy, where they put you in an MRI scanner and they target an ultrasound deep into the brain and give you a little hole in a certain strategic part of the brain. And that's been used for tremor, especially in people who have bad tremor and can't tolerate deep brain stimulation, either surgically because they're not fit enough for that sort of length of surgery or don't want the metal work. I think it's quite a good therapy. The question always is with these lesions, which was very much before deep brain stimulation, that's how it's treated. So instead of having a subthalamic stimulator, they would make a hole in the subthalamus called a subthalamotomy. What used to happen is people often, after a number of years, it started to come back. So I think when most people are looking for non-pharmacological, they're looking for physical measures. I don't really know enough about it, but there are people who've got devices which they say help. They've never really been the subject of formal trials, as far as I know, but they're certainly worth exploring.
Yeah. Great. Thank you, Roger. Are there techniques for reducing or eliminating dyskinesias?
Well, I can get rid of dyskinesias in everybody. I just tell them to stop their therapy, but that's not normally a very helpful way of doing it. So the treatment of dyskinesias is a bit limited, and that's one of the areas which I think people do need to start thinking about. So at the moment, the way to limit dyskinesias is obviously trying to smooth out the dopamine delivery, as we've already discussed, and there are ways of doing that. Apart from L-dopa, you can infuse duodopa, apomorphine under the skin and such like. The drug, obviously, which most people use is amantadine. So that's a drug which you can use to help with the dyskinesias. There are other drugs that people have talked about. So kepraleforacetam, which is a drug used in epilepsy, has been tried for dyskinesias. So there are other drugs that people try, but once dyskinesias are there, they're quite difficult to control if they don't respond to amantadine, and that's normally when people are looking at these advanced therapies, deep brain stimulation, the apomorphine and the duodopa.
The next question, it's switching around. People are voting, great. Would it be possible for someone like my husband, who's lived with Parkinson's for 40 years, and presumably has hardly any dopamine cells left, I think, is it possible for this type of person to be eligible for a cell transplantation therapy?
Yeah, so although I wasn't really talking about repairing the brain tonight, repairing the brain falls into two major categories. You either replace the cells that are there, or you try and rescue what's there. And rescuing is when you put growth factors in, fertilizers to grow what's there, and that's what GDNF, glial cell line derived neurotrophic factor, the subject of that documentary, which I'm sure many of the people here have seen tonight. Now, that therapy will never work in people who've got relatively advanced Parkinson's, because you're trying to grow back a system that's long since gone. So the analogy I always have is, if you have the best fertilizer in the world and you put it on a flower bed with no bulbs in it, nothing will grow. There's nothing wrong with the fertilizer, there's nothing to grow. So in people who've got relatively advanced disease, that type of therapy will never work. Now, obviously, replacing cells, you can do at any point. I mean, obviously, you wouldn't want to go too early because you're replacing cells and the system's already largely there. So you may damage it with your cell therapy. But advanced patients would be fine. And as I've said, people, when they first present, you could put in cells. And as long as you put in enough to deal with the deficit, once the other cells have died off, they, in essence, would have their dopamine system repaired and never need a dopamine drug, and then never develop dyskinesias and never need any of those drugs. So the sort of the hope with cell-based therapies is that ultimately they could be used to replace everything we do in the clinic now. They're not a cure because the disease progresses in other places and the alpha-synuclein pathology develops, but in essence, you would have fixed that part of the Parkinson's brain.
Good, great answer. This is a long question, but I'm going to read it out. I think it's an important one. We hear of many research projects through the media and through Parkinson's UK that raise hopes, but then they disappear. To give some examples, research on priming astrocytes to produce dopamine, developing vaccines to target alpha-synuclein. Can you update us on the current research developments, which I think you did, but I'll let you try to answer that one, because
I mean the big problem with the field is that there have been many false dawns, and it's quite easy for us to sort of think that these therapies are going nowhere, and I think what you have to do is just take a slightly longer perspective. So when I started in neurology 30 years ago, the idea that you stick a couple of wires in someone's head and attach them to the national grid would just have been laughable. But now, you know, deep brain stimulation is a major therapy. The idea that the protein would spread an alpha-synuclein could spread from one cell to another 20 years ago, people say that's madness, bonkers. But now that's been proven. The question is, can you actually silence it? So each decade, each year, we're learning new things. And in my lifetime, I would say there are several diseases where this has clearly been shown to be an incredibly effective approach, learning as you go along. So when I qualified in the mid-80s, if you had HIV and got AIDS, you would be dead in a few months. That was it. Nothing we could do. And then AZT, this therapy, came along, and everyone thought this was the cure. And actually, everyone wanted it, and then it proved not to work. But on the back of that, people learned other drugs that could work in a similar fashion. And nowadays, if you're on combination therapy for HIV, you are, to all intents and purposes, cured. Now, that was a lot of money, and it was a very clear etiological agent. You knew what caused it. But I think actually we are learning a lot more about Parkinson's disease. And I think some of the failures in the past, they have been over promoted. So people have found one thing and think they've solved the whole problem. So I think that it is prone to hype. But I think some of the issues I talked about tonight have also been a problem. So I think if you don't select your patients properly, you're not going to see a clear effect, because half the patients haven't got the condition which you're really trying to treat with your agent. So I think it has been disappointing, and it will be disappointing going forward. But I do think each year things are definitely in a better place. And if I compare to when I came in 2012, we're in a much better place with POG disease in 2021 than we were in 2012.
Great. Yeah, I think that's a really, really good answer. And yeah, we have to take the long view. The next question, I'm going to interpret it. I think the person's asking, can dopamine be produced in other parts of the body and be boosted to help with the PD symptoms?
Yeah, so dopamine, you're absolutely right. So dopamine is found in many different sites in the body. It's found in many different sites in the brain, actually. So it's not just within that nigrostriatal pathway, the bit that takes the primary hit in POG disease. So it's found in other areas of the brain. It's found in the back of your eye. It's found in your kidney. It's found in the gland next door to the kidney, the adrenal. It's found in the next to a sensor by your carotid artery, the carotid body. So there are other sources of dopamine. The problem with dopamine in the body is it doesn't get into the brain. So when Oliver Sacks in Awakenings, and they developed L-Dopa in the late 1960s, the first problem they had was they had to give people industrial doses of Sinemet and Madapar just to get enough of it to turn into dopamine to get into the brain. And so people were violently sick, would collapse with low blood pressure. So if you give people dopamine, it generally doesn't get into the brain. So even if you could boost it in the body, it won't get into the brain. Now, people have thought about it in more creative ways, and this was very much the fashion in the 80s and 90s, was to take those dopamine-producing cells from another site and put it in your brain. So people took your carotid body, they took your adrenal gland and put it in the brain. And the problem with that is they never really produced enough dopamine to make a difference. So it's a good idea, but I think the problem is it doesn't get in the brain, and the idea is to do auto-transplanting, take a bit of you and stick it in your head, never really panned out.
Yeah. The next question is about, can we test the quantity of dopamine being produced like we do insulin or thyroxine?
Yeah, so we don't have it. I mean, there are ways of doing it. So you can actually take fluid off people's back, lumbar punctures, and put it in liquid nitrogen, and you can actually do assays of dopamine. The trouble is that will give you an assay of dopamine in general in the brain, not just specifically the one site where you want to look at it. Now, in order to look at dopamine in the living brain, people do imaging. So this thing called PET imaging, positron emission tomography, where you inject someone with a particular chemical that then attaches itself to the dopamine system, can tell you something about the integrity of the dopamine system. So it's not telling you the level of dopamine like you could measure insulin or C-peptide in the blood. It will give you an image of the brain and tell you how much dopamine is in the brain based on that imaging. The trouble with the imaging is, whilst it's very helpful, it's not entirely specific for the dopamine system. So sometimes it can be a bit misleading. But so when we do trials to look at dopamine therapies, that's what we use to try and measure how much dopamine we've got in the brain. And ultimately, if we have a dopamine therapy in the brain that's working, you won't need to take an oral dopamine therapy, so your drugs should decrease.
Great, thank you. The next question is actually, I think, related to focus ultrasound again. What do you expect to see non-invasive techniques being developed to target the same area that deep brain stimulation does? And if so, when will these be available?
Yeah, well, I think the MRI focused ultrasound is the most likely. I mean, people are interested in trying to put electric currents through people's heads and do all sorts of things. So there are two things which people do that. One's a thing called transcranial magnetic stimulation, where they put a sort of a big 8 on your head and stimulate it, and that makes little electric currents in the surface of your brain. And the other one is this direct current stimulation, where they put electrodes on your brain. And I think what you can say with that, coming back to an earlier question, is some of those have been shown to work quite well for suppressing dyskinesias. The problem is they only work as long as they've been given, and they don't tend to hang around once they stop the stimulation. So if there's a way of permanently doing that without you feeling a bit odd, which is what most people seem to think when they have it, it might have potential. But at the moment, I think it's not something that we have an easy handle on how to use that and develop that.
This one might be a bit easier. It's still related to DBS. How long does it take DBS to work? And if it doesn't work, can the equipment be removed?
So DBS, so normally what they do is they put the electrodes in, then they wait for everything to settle down, and then they switch the stimulator on. And if it doesn't work, you can obviously fiddle with the parameters. So because these electrodes have little different numbers of electrodes on it, you can change the pattern of the stimulation. So it can take quite a bit of time to actually work out whether someone has a stimulator that is actually really not working. If it's not working, then the question is, have they put it in the wrong site, or have you not got Parkinson's disease? Would be the key questions. If it doesn't work, you can disconnect it. Now, whether they remove it or not, I'm not entirely sure. I know they do remove deep brain stimulators, because if people get infections, they have to remove them. But I don't know if they just fail, they just take the battery out and they just leave the wire. I don't know. But you can take, in the answer to the question, you can take the wire out if you need to.
Yeah. So our 2019 lecture was on deep brain stimulation. It's on the Edinburgh Parkinson's website if you want to view the last live lecture. The next question is about a University of York study about a nasal spray for Parkinson's. Is there any updates on this?
Yeah, I don't know about that, but I think it's on a more general question. I think trying to get better delivery of dopamine using alternative routes to oral has been very much thought about. So, as I've said, so I don't know much about this nasal one. Inhaled dopamine, we were part of a trial, and I think that's now licensed in the States to use inhaled dopamine. I think that's used as a rescue therapy. And I was talking to Tony Lang, a neurologist in Canada, and he's very excited about this L-Dopa, which can be delivered under the skin. So that may be, a sort of new therapy. So I think different routes for delivering drugs which we know work is actively being explored. And actually, I think some of them will come to clinic fairly soon.
Yeah. Maybe, David, let me know when the time is up for questions, but this is a really good one. It's about the CDNF trial that's been conducted by Herantis.
Yeah.
There was a positive outcome of phase 1/2, but believe the delivery method is not the way to go. They're working on a nano version. Can you comment on this?
Yeah, so CDNF, for those who don't know, so the GDNF study, which many of you guys would have seen in the trial down in Bristol, used a rather complicated delivery system, something like out of the Matrix, where they had a sort of little port behind the ears, and that would then feed into several catheters that went deep into the brain. And the group in Scandinavia developed a sort of a different treatment, which, as Thilo says, CDNF, which works to sort of rescue cells. It's not a pure growth factor, it's to sort of rescue cells in many different ways, and it was delivered through the same system. And my understanding is that the data showed that it may have some benefit, but there were certainly complications with the surgery and the equipment that was used. So I think if these types of therapies are going to become more mainstream, the delivery into the brain is going to have to be much simpler than the complicated neurosurgery that's required to put it in, and also the extensive metal work and catheters that you still have in your brain afterwards, because that is an infective risk. So I think, and people are working on that. So delivery devices for therapies, people are actively working on, and there's cross-fertilization from the brain cancer field into that, because obviously a lot of people who get brain cancer, now they put catheters and inject things into it. So I think there will be better delivery systems, but the ones that exist at the moment, whilst they're very clever and work in expert hands, are probably not the way forward.
Thilo, if Roger's happy to go on for another 5 minutes, that's fine.
Oh, good. Oh, good. Yeah, because I was going to ask a really nice question, but it wasn't the top voted. I'll ask a few more. The next most popular question: sometimes the dose doesn't seem to have an effect, but when I exercise, it can kick in. Can there be some science behind this, and can you report on the cue for kicking in?
Yeah, well, there must be a science behind it. I don't really know it. I mean, often it works the other way in the sense that, you know, if you're trying to absorb things from the gut, you need blood flow through the gut. And if you do exercise, the blood tends to go from the gut into the muscles. So you can imagine that it might make it slightly worse. I mean, it may be that the answer is that when you exercise, of course, you stimulate a lot of your own adrenaline, noradrenaline, and all of these agents which activate your what they call your the sympathetic nervous system, and that in itself has benefits. But if I'm honest, I don't know the answer. I could make up an answer, but I don't know the answer, I'm afraid.
Well, it's a good question, but I remember hearing studies of rats when they exercise, they release neurotrophic factors, but that's, I guess, a slow way of rebuilding.
Yeah, I mean, BDNF, this growth factor, as you say, brain-derived neurotrophic factor, that is increased by exercise, but that won't give you an immediate kick when you're exercising your drugs. It must be something to do with the fact the drug gets taken up better through the gut and into the blood-brain barrier. And whether it's something to do with the sympathetic system opening things up, I don't know.
So the next is a really good one. If inflammation is a big part of Parkinson's, should we not be trying immunosuppressants to treat the condition?
Yeah, well, I think we should. Well, I don't. So I would. First thing I would say is don't go out and start yourself on any immune therapy or anti-inflammatories, because this is unproven. But that is exactly what Caroline's trying to prove next door, that if you give immunosuppressions, which actually target mainly the peripheral immune system, by suppressing that, you will actually have better brain health and your disease will progress less quickly. That's the theory, but we've yet to prove it.
Why and how does PD, the lack of a neurochemical in the head, give rise to fatigue? Is this something to do with mitochondria, and what can be done to offset this?
Yeah, so, well, I would say there are probably two reasons. I mean, again, it's a very complicated question. So I think there are two reasons, really, I think, with it. One is that I think if you're working with a disadvantaged nervous system and a motor system, everything requires more effort. And fatigue is to some extent related to how much your brain has to be activated to get you to move. So when you send a signal down from your brain to your muscles, a part of that signal, a copy of it, goes into another part of your brain and says that's how much effort you need to move. So obviously, if everything's a bit slow and stiff, it has to put more down that pathway. So the message coming sideways is that's more effort. So that's why everything is harder work, which of course makes for an element of fatigue. It's also fair to say that dopamine's very heavily involved with motivation and drive. And so I think part of the reason people get fatigued is there's an element of sort of apathy and amotivation. You know, it's difficult to get going, so everything's harder work. I don't think that's the whole answer, because I think there are other factors that play into that. I mean, sometimes the drugs, especially the dopamine agonists, so the rapinorols, pramipexole drugs, can make people feel tired and sleepy. And Dave Breen will be doing work, I'm sure, on sort of sleep and you know, if you don't sleep properly, that makes you tired anyway. So I think there it's multifactorial, but I think those are some of the reasons why people feel fatigued.
So next one is an interesting one. So I'm happier to see the name Parkinson's than without the word disease. Is it a disease because people are affected in so many different ways? Could it be termed a syndrome? It would make me feel more positive.
Yeah, I think you can. It's an interesting one, really, because I was thinking about this the other day, actually, because I was thinking about cures, and I was thinking about asthma. And asthma, we don't really cure people with asthma. We treat them, and we treat them very effectively, but we don't call it asthmatic disease. We do just call it asthma. But it is a disease because it's inflammation of the airways, which gives you a syndrome. So, I mean, people have spent a lot of time debating this. I suppose part of the problem is when you're as old as me and you've got so used to calling it Parkinson's, you call it Parkinson's disease. The trouble with Parkinson's syndrome is it starts to embrace things which clearly are not Parkinson's disease, if you like. There are a whole series of Parkinsonian syndromes which are well known, which have a different pathology and a different course and such like. So I think there's a worry if you start using the term Parkinson's syndrome that they start to get lumped in, and it makes it harder to design trials and get the right patients for the right therapy. But I take the point that I think disease often is not a particularly nice term, and perhaps we should just talk about Parkinson's and leave it at that.
Great. I'm going to ask the last question in a moment, but before that, I wanted to highlight a post that I did on the question line about another patient online event called Parkinson's and Huntington's Clinical Trials: The Participant Experience, and I'll put the link in the chat, and that'll be on November 9th. So last question, Roger. Why and how do you personally keep going when there are so many challenges with this research? What motivates you on bad days? Thank you for all you do, Roger.
What motivates me is the patients, really, and my colleagues. I mean, one of the things is I give these sort of talks, and it makes it sound as though, well, I hope I haven't. It's the cleaner. I hope I haven't made it sound as though it's all my work. 'Cause most of it isn't my work, but actually, it's working with people to solve problems. And the amazing thing is, I personally love people, and I love patients, and I always want to help them. And if I can help in any way, and this is one way to help them, then that's a terrific thing to do. And a lot of the things we end up doing come from what people tell us. So that's why, when I was saying, what have you got to do if you've got Parkinson's? Get yourself to a research clinic, get yourself to someone who's interested in you. And that's a big start to help you with your condition. And it helps us.
Super, super. Thank you. Thank you. I'll let David take it from here. Thank you very much, Roger.
Thank you.
Yes, thanks, Roger. Some great questions and some great answers as well. Thank you. We've had 999 people register for this year's Edinburgh Parkinson's lecture. I kept the registration open to try and make 1,000, but we just missed out by one. 65% of those who've registered either have Parkinson's or they're the family member or carer of a person with Parkinson's. We've also had lots of healthcare professionals registered and a number of university academics and students. This is precisely the right audience, the same audience as the people who inaugurated the Parkinson's lecture 10 years ago, Patrick, Mark and others. Precisely the type of people that they wanted to bring together to engage. The only difference now is that through the internet, we're able to do it with a lot more people. We all owe Roger Barker a great vote of thanks. Roger said that when he was in Edinburgh last time, he was presented with a very nice bottle of whiskey. Here's a bottle of whiskey, Roger, but I'm afraid it's a virtual presentation rather than actually something for you to take away. But it's the thought that counts. We've heard a really inspiring lecture from Roger. This is his second Edinburgh Parkinson's lecture. And if the Edinburgh Parkinson's lecture is still running in another 10 years' time, I'm sure Roger won't be too decrepit to come back and deliver the 20th anniversary lecture. I'd like to thank my colleagues who've helped with the presentation today, Patrick, Mark, David Breen, and Thilo for running such a great Q&A session. I'm also grateful to the staff of the Royal College of Physicians of Edinburgh for their help in putting the presentation on. I'd like to end by encouraging all of you to consider making a donation to Parkinson's UK to support their work to try and find a cure for Parkinson's. If you look at the very top of the web page, you'll see a Just Giving link. If you go to that link, you can make a donation. If you're there and you're asked to add a message, please type Parkinson's lecture into the message box. That will ensure that any money that you do donate will be used purely for Parkinson's research. I hope, we all hope, that you've greatly enjoyed this year's Edinburgh Parkinson's lecture, and that many of you will consider joining us again for the Edinburgh Parkinson's lecture 2022. Thank you. Bye-bye.